Okay, so it's a pleasure to introduce our last speaker, Dr. Lee Rubin. Dr. Rubin is professor of stem cell and regenerative biology at Harvard and the director of translational medicine at the Harvard Stem Cell Institute. And I think those titles really reflect how Lee straddles the worlds of basic biology and translational medicine. Dr. Rubin, I think, is one of a small group of scientists who have managed to achieve success in both industrial and academic settings. He was the CSO of Curis and discovered and developed an inhibitor of sonic hedgehog, which is currently being used in the clinic today for the treatment of cancer. He then went on to Harvard and brought with him high-throughput drug screening technology that he's used not only for drug discovery, but also applied to understanding disease mechanisms and identification of new targets. So with that, I want to thank Dr. Rubin for coming out here to the West Coast.
Well, thanks to, do I have to turn this on? It's on. It's on. Thanks to Anne and Luke. It's a pleasure to come out to the West Coast, I can assure you. So by way of disclosure, but not financial disclosure, my talk is completely unrelated to the two previous fascinating talks. If you cast your mind back to very early this morning when Alison Motry was giving his talk, my talk is most related to that one. Also, by way of disclosure or excuse, my talk is going to use iPS cells as its basis. It's going to talk about disease and problems that we've addressed using iPS cells. It's not really going to talk much about high throughput anything today. I'm going to ask a few questions, which are simple questions, I think, and try to answer them for you. The answers came from studies of iPS cells, and I will say could only have come from studies of iPS cells. And at the end of my talk, there's going to be a little twist, which I hope people find interesting. Okay, so this you've seen now 150,000 times before. It's for all of us who have been interested in modeling disease, as Anne said. And just a few years ago, in fact, the last time I was speaking here, it was not so commonplace that people really recognized the potential value of using stem cells not for therapy, but more for modeling disease. And that's my focus today. Now, if you're a neurobiologist like I am and you want to study diseases, there are a lot of diseases of the nervous system that you could consider studying. Some are easier than others. So I've tried to sort of put in green the things that make diseases easy to study in an iPS format, and in red the things that make them harder to study. So for those pesky psychiatric disorders, they're relatively early onset, especially autism. So that's good when you're trying to model diseases in vitro. But they're really highly polygenic, which is bad because nobody knows how many forms of these diseases there are. In diseases like ALS, Parkinson's, and Alzheimer's, the classical neurodegenerative diseases, that's all sort of all red. They're highly polygenic. They have a strong non-familial component. And they're and they're late onset, really late onset. And the later the onset, the harder it's going to be to model them in vitro for obvious reasons. So I'm not going to talk about any of those today. I'm going to talk about one that has some advantages, much as Alison introduced to you this morning, which is a motor neuron disease called spinal muscular atrophy. And this is monogenic, early onset, so green. But it's going to have some little kind of unexpected aspects that I'm going to try to explore with you today. Okay, so this is spinal muscular atrophy. It's caused virtually all of the time by a deletion in a gene called survival of motor neuron 1, which is a gene that mediates RNA splicing. But I'll tell you a little, it's not really understood how it supports motor neuron survival in spite of its name. I'm going to tell you how it does that. At least to some degree. 2% of people in the world carry only one copy of SMN1, and about 1 in 5,000 live births have children that have 0 copies of SMN1, and then they will have spinal muscular atrophy. This disease exists in different degrees of severity, ranging from type 1, the most common form, generally fatal before kids are 2, to type 3, a much milder form of the disease. And what determines the severity of the disease, it is thought, is pretty much the number of copies you have of a duplicated gene called survival of motor neuron 2. Survival of motor neuron 2 codes mostly for a truncated protein which is rapidly degraded, but 10% of the time it codes for the same protein product as SMN1. So if you have enough copies of SMN2, you can be more like type 3, but if you have few copies of SMN2, you'll be type 1 and not survive very long. So that sounds, as these kinds of diseases go, pretty darn simple, but of course, one of the unappreciated aspects, or less well-appreciated aspects, is kids with the same number of SMN2 copies can have vastly different forms of the disease. So some can have forms of the disease that are more like type 1, some can have forms of the disease that are more like type 3, suggesting that there may be other disease-modifying genes. One of the canonical aspects of this disease, it is asserted that you can, if you're a parent of a kid with this disease and have one copy of SMN1, you are completely normal. I am going to challenge this at the very end based on some work that we've done. That is the little twist that I want to talk to you about. But it is assumed in the field that this is true. Given all this, even though I've raised these complexities, the relative simplicity and the fact that there are drugs now in the clinic, including one developed in San Diego by Ionis and Biogen, it's not really known why motor neurons die in this disease. And this is a common problem when people study neurodegenerative diseases, which tend to affect some cell types more than others. Why those and not the other cells? Why? This is a ubiquitously expressed gene. All cells experience reduction in SMN levels, and yet, in this disease, SMA, it seems like motor neurons are more sensitive to the loss of SMN than other kinds of cells. So why is that? That's number one. And then there's another little kind of unexplored aspect of this disease, which is that it has two phases, kind of an acute phase and then a chronic phase. So there's a period in which children lose a lot of motor neuron function and a lot of motor neurons. But after that, the disease enters this chronic phase where the progression of the disease, at least from a motor neuron perspective, really slows down. And I will kind of make the argument that SMA is not a motor neuron disease when the kids are in this period. Okay, so that's another little prediction, and I'll explain why I think that. Okay. All right, so in this world, and when I should also say, there's a pretty darn good mouse model of SMA, and most people who have studied this disease have used that mouse model virtually exclusively, and it's sort of not been, the penetration of human iPS-derived studies has been less in this field than in many other fields because of the presumed quality of the mouse model, which I'm not sort of debating. Instead, I'm going to talk about, in spite of the good mouse model, things that we've learned about the disease that relied on the ability to make lots of human motor neurons. Okay. So in particular, these are the two questions I'm going to try to answer for you. How come motor neurons die and other cells don't in the early phases of this disease? And why do only some motor neurons die? There are survivor motor neurons. What is the distinction between those that die and those that live? So that's what I'm going to try to answer for the bulk of my talk. But very, very quickly, because I'm just going to go over how you did it. So this is what I'm going to call the classical use of iPS cells. You have a disease. You try to reproduce it in a dish and study the disease in an easy-to-control setting. So the second part of my talk will be the exact reverse direction. So basically, without showing you all the data, I'll show you a little bit of it. We were able to reproduce the disease, i.e. we could get SMA motor neurons dying in a dish. We were able to show that the motor neurons are kind of predictive of real human response in that we tested the two drugs that are in the clinic now and showed that they worked on these cells very well to to increase the level of survival of motor neuron protein and increase the survival of motor neurons, whereas many other compounds that have failed in the clinic actually don't work in these cells. So it does have some sort of preclinical, in this case, post hoc predictive analysis. We also have carried out small molecule screens using these cells, which I'm going to quickly show you a result that was derived from them without showing you the actual screen itself. But the first thing I'm going to tell you about is why motor neurons die business. Okay, so we made we derived iPS cells from lots of SMA patients that had different severities of the disease, the really severe ones, the really mild ones, and we made motor neurons from all of them, and it's possible to make motor neurons. And then we studied them. We first showed, again, I'm showing just a hint of the data. This is actually published. Motor neurons in these cultures produced from patients die, whereas other neurons that are in the same well, other kinds of spinal cord neurons, do not die, suggesting that the major feature, the major well-known feature of SMA, i.e. motor neuron death, can be recapitulated in vitro. And we showed also that the severe patients gave rise to lots of death, and the milder patients gave rise to less death, and so on. So at this very simple level, we had a disease model. Now, that was when we started to ask why the motor neurons die and the other cells in the well don't die. So what was different about the motor neurons as a population from the other cells? Well, these are very heterogeneous cultures, so we relied on a method that you've heard, again, early this morning from Tracy Tran introduced, and it was a method that was developed in Doug Melton's lab and then also used by Paula Arlada in my department, which is called MARIS, Method for Analyzing RNA Following Intercellular Sorting. For those of us like us neurobiologists who don't necessarily have cell surface markers that allow us to purify cells, it's a method by which you can fix and permeabilize cells, stain them, for instance, with transcription factor markers, which for many kinds of neurons are known, fax purify the cells based on the expression of these intercellular markers, and then do RNA sequencing. And so that's what we did. Needless to say, we found differences between SMA motor neurons and control motor neurons. And one of the things that we followed up on that came jumping out of this analysis was there were lots of genes that were dysregulated in the SMA motor neurons, which indicated an activated ER stress response. For instance, this shows activation of a transcription factor, ATF6, that's sensitive to ER stress in the nuclei of motor neurons, but there are lots of other neurons in this culture, and so it appears to be mostly activated in motor neurons, even though all of the cells have lower levels of SMN protein. So it appears that one thing that happens is, for whatever reason, and I'll tell you partially a reason, motor neurons, when they have low SMN, activate a prolonged stress response pathway, which leads them to die, whereas other neurons actually don't activate that same stress response. And you can produce this same thing if you just take wild-type motor neurons and reduce the level of SMN with an siRNA, you get the same thing, activation of a stress-induced pathway in motor neurons. We went on to show, using a variety of genetic and also chemical inhibitors of the stress pathway, that you could rescue motor neurons even though they had low levels of SMN. You could keep them from dying by just reducing the level of ER stress. And that was true in human motor neuron cultures, and it was actually true in the mouse model of SMA. If we injected guanabens, in this case, which is one of these stress relievers, into mice, we showed that you could preserve many more normal neuromuscular junctions in the mice, prevent the motor neurons from dying for some time, and keep the mice alive for a little bit longer. Partly the reason motor neurons die and other cells don't, even when they all have low levels of SMN, is because motor neurons experience more stress than other neurons do under these settings. Well, how come? Well, one of the things that seems to go on in motor neurons is they actually misprocess chaperones. Now, this was kind of interesting because most of you know that most of the, at least, neurodegenerative diseases are characterized by protein aggregation. And when cells have protein aggregation, they typically have some screw up in their chaperone pathway and also some ER stress response. But this is the one disease that's caused by loss of a protein, and it was not thought to be associated with protein aggregates. That is also not true. They actually do have aggregates. We're not sure yet which proteins are aggregating, but they do have actually aggregates building up, even though it's an early onset disease that was not thought to be associated with aggregates, and that's part of why that happens. This is interesting kind of in another way. I'm going to come back to this because it suggests that SMA, a motor neuron disease of genetic origin, early onset, and ALS, a motor neuron disease of very different origin, late onset, share a couple of similarities in that both are associated now with ER stress, and of course both are characterized by protein aggregates. So I'm going to come back to the similarity between those two diseases soon. Okay. So now the second thing I want to deal with is why. So in the cultures, motor neuron cultures, not every motor neuron dies. Just like in patients, not every motor neuron dies, and just like in mice with SMA, not every motor neuron dies. So how come? Well, could be death is just random. Some die, some don't die. There's a certain probability each cell will die, and that's the way it is. Or it could be there are actually molecular differences among the motor neurons such that some live and some die. So which is it? So actually, we've known for years under many, many settings, the cultures are very heterogeneous from an SMN per cell point of view. So even though I'm not showing you this stuff, we do a lot of kind of phenotypic screening where we're looking for small molecules that modulate SMN levels. So we do a lot of high-throughput antibody staining of cells. So we've collected about 10 terabytes of images of motor neurons stained with antibodies against SMN. And suffice it to say that we have never seen a culture which this didn't exist. There are within a one single microscope, and this just, by the way, is not just for motor neurons. There are some motor neurons that have high levels of SMN. I'm just using an arbitrary descriptor. There are some that have a medium level of SMN and some that have a low level of SMN, just as near neighbors. Well, since, and I should say, this is, having measured lots of these, you can see all motor neuron cultures from patients, from mice, SMA mice, but even control cultures have a distribution of SMN levels per cell. And in fact, there's kind of an overlapping distribution. So SMA motor neurons have lots of cells that have low SMN, as you'd expect. But there are some that have sort of normal levels. And wild-type cultures, which of course there's also some cell death, have, mostly higher levels, but there are also some with low levels. So, and this is true in all cultures. So what does that suggest? Well, since SMA is a disease caused by low levels of SMN, since some cells in the culture have low levels of SMN and some have high, could it be the case that it's actually the cells that have low levels of SMN that die preferentially? So that also appears to be true. So we've done this in many, many ways. If you look at which cells in the culture are dying, they tend to be the cells with low SMN. But it suggested an experiment that we did, which is just to say if you increase the expression of SMN in whatever means, in this case it was done with a lentiviral vector, we predict you should, by shifting the average amount of SMN, be able to keep motor neurons alive. And that is also true. And it's true in wild-type cultures, and it's true in SMA cultures. Now, it's also interesting true in ALS motor neuron cultures. So first of all, so we made motor neurons from a variety of ALS iPS cells. They also have heterogeneous distributions. And excitingly, at least for us, it is also true that if you increase SMN levels in ALS motor neurons, it shifts the SMN distribution, so you have fewer cells with lower SMN that have a higher probability of dying. So increasing SMN promotes motor neuron survival, full stop. It promotes motor neuron survival of SMA motor neurons, but also of any motor neuron put under a stressful condition, even if you put toxic compounds on motor neurons. There are some that survive, and they are always the ones with higher SMN. So SMN just appears to be a motor neuron survival factor. Okay, so to summarize that, we feel we've kind of modeled this disease, at least to some degree, and we've been able to take advantage of our ability to make lots of motor neurons, so we have mass, and we also our ability to track the behavior of individual motor neurons, so we can do things at small scale and large scale to try to begin to answer a couple of questions that were just not well understood in SMA. That is, why do motor neurons as a population die, and why do some die and others not die? Importantly, if it's true, if what I said is true, and ALS and SMA are actually surprisingly similar, and increasing SMN could be therapeutic for both, that suggests that one drug of the right kind might be able to treat both motor neuron diseases, which is somewhat surprising. But interesting if it's true. Okay. So now in the next few minutes, I'm going to talk about this other way of thinking about the system. This is dish to patient. So I said patient to dish. We know something about a patient. We try to reproduce the disease in a dish and try to make the dish match the patient as much as possible. But given that we can make these cells, could we learn anything about the patients that we didn't know by studying their cells? Okay. So what we observed, and we've observed this for quite some time, was that the iPS cells that we made from these patients themselves are aberrant. And in particular, they have a kind of a skewed balance between proliferation and differentiation. We had already found that other types of stem cells, like muscle satellite cells, also behave aberrantly. In fact, Monica Hayhurst-Bennett, who's in the room, who now works at Genea Biocells, was the postdoc in my lab who showed this. So SMA is called a motor neuron disease or a neuromuscular disease, but everybody thinks the problem is the motor neurons. But it's very clear there's an independent defect in muscle, and it has to do with abnormal ability of muscle satellite cells to form muscle because they differentiate prematurely in this particular case. So basically, by a variety of studies, what we proposed from these IPS studies was that patients would have defects other than neuromuscular defects. And I'll show you why in a sec. And this has now been confirmed, by the way, in patients and in also mouse models. And we're also predicting that parents are not completely normal in ways that I'm going to show you, and we were able to, at least tentatively establish this in a kind of fun study that we just recently did. Okay, so here are, we've looked at iPS cells now from these patients for quite some time, and I'm just giving you just a very quick analysis. So this is a nanostring analysis of iPS cells derived from controls or SMA patients. And basically, they're skewed in terms of their differentiation. Interestingly, and we've studied this in some detail, they're skewed towards the generation of neuronal phenotypes. In fact, they make motor neurons better than control ones. They just die faster. But if you look really carefully, they appear and then they disappear. And they make gut tissue, endodermal tissue, really badly. And so this suggests, again, broader kinds of defects than one would have imagined. It also suggests that, which we already knew, that different kinds of cells respond differently to reduction in SMN. So we know motor neurons are the most sensitive to the reduction in SMA, such as seen in the kids, but this is suggesting that there might be other tissues that are sensitive to smaller reductions in the amount of protein over a longer period of time. In other words, maybe the parents aren't as normal as everybody thought. To investigate that, we collaborated with the bioinformatics department at Harvard Medical School and obtained medical records from about 50 million people. We queried the databases for kids with SMA, and then by definition, their parents are both carriers. So we then examined the medical records of the parents and compared them to age and sex matched controls, and also to controls where we included parents of kids with another childhood disease, cerebral palsy, to take into account things that might happen if you have a sick kid. And it was kind of interesting to us because they clearly have their issues. We've artificially, just for the purpose of this, divided their issues in the kinds of things that kids themselves have, like neuromuscular things. But they also have other defects as they get older, in other tissues, like bone, as an example, is a big one. They also have tremendously increased predisposition to having epilepsy. Okay, so this is really, really interesting for a couple of ways. Number one, because it does suggest the possibility, and you can learn stuff from this kind of IPS approach that you could not, well, you could have done this, you could have done what we did anytime, but we wouldn't have thought of doing it if we hadn't been looking at these iPS cells. And we think that this is important for a couple of interesting things, but in particular, whereas I already hinted to you that I think SMN-elevating drugs might be used to treat ALS on top of SMA, this suggests that parents have defects that might also be treated with drugs that increase SMN levels because their defects are due to a lower level of reduction playing out over a longer period of time, if that makes sense. Okay. So finally, just to thank the people in my lab who did this. So Xianeng did all of the RNA sequencing. Nadia and Natalia did the heterogeneity analysis of SMN cells. Maureen did the IPS study, and Scott Lipnick and Nina Makhartova did that kind of informatics stuff in collaboration with Zakahani at Harvard Medical School and Basil Darras, who's our pediatric neurologist who's helped us with this. Thanks very much.
Awesome. As always, really great talk. I have one question. I think I heard this before from you, but what is really the cause of this heterogeneity on the levels of the SMA protein?
You know, it's a great question, and it's been a pesky problem to try to answer. The one thing I can tell you is there's a lot of post-translational regulation of the degradation of the protein, absolutely for sure. And there are a number of signaling pathways like neurotrophin signaling, for example, and levels of calcium, intracellular calcium, that regulate the level of the protein. We know that, like you were showing activity patterns this morning, and we know that there are different levels of activity in the different neurons in the culture, depending on the number of synapses that they happen to make as they're forming in culture. So our hypothesis is the more active ones have higher levels. Now, in vivo, the two pathways, the neurotrophin pathway, another extension of this hypothesis is even during developmentally programmed cell death, it might be the case that SMN is a regulator of motor neuron survival, and their neurotrophin signaling and activity play out as important regulators. So that's our best guess at the moment. But it's just been, we've tried to follow the activity of individual cells. It's sort of just been a tough game. We finally have been able to label functional SMN fluorescently, so now we can prospectively look at the cells and ask those kinds of questions. But it's taken us quite some time. Yeah.
Do the read-through compounds, the SMA compounds, do those work in ALS-derived?
No.
So is there any?
I'm glad you asked that question because I didn't want to badmouth it. Well, it's not. I mean, because they only work where there are SMN mutations. They don't increase the protein unless there are enough copies of SMN2 in the absence of SMN1 mutations. So they basically work primarily on lots of copies of SMN2, and they won't work as well if there are normal SMN1s. The compounds we've looked at regulate the rate of degradation of the protein. They work just across the board in every kind of cell because they work on the truncated and full-length protein.
That makes sense.
Yeah.
Yeah.
I was wondering if you looked at the transcription or the transcriptome of the fibroblasts from which you made the IPS, and then see if any aberrant expression was carried over into the IPS cells.
You know, we haven't ourselves yet directly compared the fibroblasts to the IPS cells, but it's a great thing to do, and we certainly plan to do it. Yeah.
Because it could be carryover.
Absolutely.
I have a question. The SMN sounds the disease was caused by mutation. It's pretty simple. Besides, I mean, so far I just asked about the therapeutic area. So far, besides use the elevate the SMN protein level as a therapeutic approach, do you know any successful case for use stem cell to treat SMA?
At the moment, that hasn't been. There's a gene therapy vector in the clinic. There are 3 splicing modulators in the clinic, but no one yet has proposed cell transplantation for SMA anyhow. For ALS, people have thought about it. You know, motor neuron diseases are problematic because they're so widely disseminated, and presumably you'd have to inject a lot of motor neurons up and down the spinal cord axis, so it's maybe not the preferred approach. Yeah, well, in ALS, I think there are defects of both cells are aberrant, for sure. In SMA, I'm sure at the end of the day, if you look carefully enough, the astrocytes will turn out not to be completely normal either. But it's a good point. And just for what, a lot of these things are historical because the splicing defect in the SMN gene was early recognized. That's been people's focus. And sometimes it's just what, it's like the lowest hanging fruit, and I think people, and it's been effective. As I say, there are 3 things in the clinic, including something in phase 3 at the moment, which is a splicing modulator. So I don't want to pretend that the efforts haven't been successful. They have been successful.
Okay, thanks very much, Lee, and thanks to all the speakers for the session.